Hello. Welcome to the York College first annual Student Research Day. And I'd actually like to begin by just asking that we applaud for all of our combined efforts. I've learned a lot of things over the... my name is Professor Rishi Nath, and I'm actually an assistant professor in the mathematics department, and recently was asked by the provost, Provost Griffith, to direct a new Office of Undergraduate Research, which is really a delight and an honor for me to do. The main event for this first semester, the inaugural semester, is this event, the first annual Student Research Day. And so in working over the last few weeks with the team that the provost put together, really a wonderful team, including Larice Miller, Holger Henke, assistant provost, the provost himself, Dean Melatius, Joanne Bob, and many, many others. We really put together something, we put together the framework of something that allowed all of you to really fill in the blanks in a really big way. And so when I was thinking this morning about what I've learned over the last few weeks working on this project, and really a couple of months, but really intensely over the last few weeks, I've learned that shea butter works well to reduce the appearance of shadows under your eyes after 5 hours of sleep. That was one thing I learned. It was sort of a remarkable discovery. I've learned who to talk to book a strange section of the second floor that doesn't have a name. And those are some of the things that I've learned. But I've also learned that there is a remarkable amount of research that is going on at York College. And that's why I asked for us to have that round of applause. Furthermore, I've learned that there is a lot of interest in research and creative works. So even those of you who have not yet worked on your project or mentored a student have said to me, that's something I really want to do, and I've been waiting to do it. So that has been very enlightening for me, and I'm very enthusiastic about that. In introducing the day and welcoming you, I have to say a few words about the vision for student research as a part of the undergraduate experience. Certainly, it has not always been the case that asking undergraduates to do research is a part of the undergraduate experience. In looking up some of the history of undergraduate research through the resources of the Council for Undergraduate Research, I found that undergraduate research started in American liberal arts colleges in the 40s and 50s. When the Soviet Union put a dog into space, that's right, before a man was in space, there was a dog in space, and that was the Sputnik mission. The American government got really nervous and decided that they better fund more undergraduates to do scientific research so that we could put dogs in space too. And you know, as a result of that, it developed and developed, and now we're at a stage where in many disciplines, not just science, undergraduate research is thought of as something that's really worthwhile as a part of the pedagogical experience. And while York is in many ways trying to find its way to the national level of what other colleges are doing, in many ways we're leading at CUNY. And Provost Griffith really had this vision of what a small undergraduate institution like ours can do to support people who are integrating these kinds of experiences, as well as to facilitate new experiences and to reward people for engaging in them. And I'm very happy to be part of it. So I wanted to just say a few words about that. And secondly, although I'm going to introduce Provost Griffith, who will introduce Dean Furton, who's our keynote speaker, I did want to say that while I was driving in this morning, I thought to myself that it was, and I actually shared this with Dean Furton, that it was a very nice choice to have Professor Furton, because of the fact that while he has a lot of experiences, he comes from the chemistry area. That's his specialty. And at York, as you can see by the posters, the chemistry department is really doing a very nice job and as is the biology department and several others, of sort of giving us a model for what undergraduate research might look like. Furthermore, Dean Meletius is a chemist, and we have this wonderful collaboration with the FDA that Professor Chakravarti is leading, who's also a chemist. So the chemistry theme is really one that we can kind of take as a model and move into other areas. And so I really commend Provost Griffith on that choice. So with no further ado, I'd like us to have a warm round of applause for our Provost, Ivala Griffith.
You can give the Provost a better round of applause than that, can't you? Good afternoon. Good afternoon. Good afternoon. Good afternoon. Good afternoon. Good afternoon. I have an envelope in this hand with a $100 bill in it. I'd like to have someone get this envelope. However, I didn't say but, however, in order for you to get this envelope, you've got to complete a sentence as a test. I'll read you the first part of the sentence, and you will complete the sentence, and whoever completes the sentence correctly first, and I spot you, you will get this envelope. So here's the beginning of the sentence. Not everything that can be counted counts. I didn't see anyone standing up. Not everything
that counts can be counted.
Oh, I forgot to say only students can get the envelope. Sorry. Sorry, Professor Curry. Sorry, only students. But since the faculty got their sentence correct, and it's one of my favorite quotes from Albert Einstein, I want to give something to the student back there who I spotted who identified the rest of the sentence. Can you come up? Give her a round of applause. Tell us your name, tell us your major, tell us who is your mentor professors.
Okay. Hello, my name is Stacey Ruiz. I'm a psychology major, and my mentor is Professor Pires, who's sitting right there.
So we're going to give an envelope and an IOU to Professor. But we'll give you a book.
Okay.
A book that you're going to find useful in the rest of your undergraduate career. And when you graduate and move on, whether that moving on is to graduate school or to the world of work, you're going to find this book useful. It's a book by Kate Turabian, a manual for writers of research papers, theses, and dissertations.
Thank you.
Now, not everything that can be counted counts, and not everything that counts can be counted. And in college, you can, thanks, Luis, I turned it over deliberately so that no one would see Albert Einstein. You know, in college, we can count the number of courses you do. We can count the number of credits you take. But for me, more important than the courses and the credits is the value of the knowledge you get from those courses and credits. It's really the value of the knowledge that counts, that matters. We can also agree that in college, it counts to go to class. But it is not only the classes that count. It's not only the classes that matter. Picking up on an E word that Professor Nath used, for me, college is about a variety of experiences. And one of those experiences that is fundamental to helping you prepare for the next stage of the journey, whether that next stage is graduate school or professional school or the world of work, one of those experiences is research. And as we heard from Professor Nath, research is not only in the sciences. I'm anxious for all of our students to be able to embrace a research experience, even if you are in the humanities. And you will notice that the epigraph that I used from the writer, that is one of my favorite writers from the Renaissance, talking about research, she is not a scientist. She's an anthropologist. She was a literature person. Research exists. Research can be reflective in the experiences you get in college in any discipline that you're part of. So we've got an opportunity with this undergraduate research program to ensure that the things that count, the things that matter, include research, because those things are going to help you be more competitive when you are finished with York, competitive whether you want to go to law school, to do the PhD, to do other things, or whether you want to go directly into the world of work. It is not only their classes that matter. Research is one of the things that counts. And in thinking of a person who we would want to invite to be the keynote at this inaugural Student Research Day, I thought of Professor Ken Furton because way back when I was at Ken Furton's institution, one of the things that impressed me, I was then Dean of Budget, Associate Dean of Budget in Arts and Sciences. Ken Furton was one of our faculty in Arts and Sciences who, in spite of the fact that many of us had PhD students, Ken Furton never neglected his undergraduate students. He may have had postdocs, he may have had doctoral students, but he also had undergraduates in his lab. And when I went back to Florida for a football game, I said to Ken, "Are you still doing stuff with undergraduate students?" And he said, "Yes." I said, "I'd like you to come and talk a little bit to our undergraduates about the extent to which your experience in undergraduate research as a student, and then your experience in pushing the horizon of other human beings who are students at their stage of life, is something that our students and faculty can profit and benefit from." I'm not going to read you the bio of Ken Furton, it's in your program, but you can see that not only is Professor Furton someone who is an accomplished researcher and publisher, securer of grants, but he's also able, even as a dean, to continue teaching and researching, helping to make us appreciate the importance of not having to give up your research if you're a dean, and he did not give up his research when he was a department chair. It is possible for us to balance in the academy a variety of contributions we make, whether leading a department, leading a college, but not abandoning that important experience of research, whether undergraduate or else. And so it's my pleasure to invite colleague and friend Ken Furton to share with us how undergraduate research can be part of the experiential landscape in college, helping us not only to take the classes that are important, the classes that count, but understand that college is much more about things other than classes that can complement the classes. Not everything that can be counted counts. Not everything that counts can be counted. Undergraduate research counts. Undergraduate research can be counted. Thank you, Ken.
Thank you, Provost Griffith. I want to start by thanking all the organizers of this, this wonderful event. I looked out there and saw a tremendous number of posters. I mean, Florida International University is now, at least we like to consider ourselves a major research university, and we would probably have a hard time pulling off as many posters as you have out there. So I'm looking forward to coming out and sharing and talking with the students and seeing what's happening. So I want to thank the provost and all the other members of the administration and the faculty for inviting me here. And I always have way too many slides, so I'll go through them very quickly, but most of them are just to jog my memory when I'm trying to decide what to talk about. So Provost Griffith is not only a friend of mine, but also a role model, because I'll tell you, when I became chair of chemistry a long time ago, I started at FIU back in 1988, and worked my way through the ranks as graduate program director, helped start the PhD program there, and then one half of the chemistry department wanted me to be chair, and the other half of the chemistry department didn't want me to be chair. And I was told, well, if you do that, it's going to kill your research, because I was a new associate professor. And I sort of took that as a challenge. I like being challenged, and so I continued to do research while I was chair. And in my second term as chair, then Dean Griffith got tapped to move to the Honors College to run the Honors College at FIU. And he recommended I take his position as Associate Dean of Budget. And I wouldn't have normally considered it, because what I saw in the Dean's office at the time was mostly just a lot of paper pushing and not very interesting work. But Provost Griffith was always still continuing to do research, working with students, continuing to publish books. And so I saw that if he can do it, maybe I can do it. And so I did do that for 6 years until 3 years ago I was tapped to be Dean of the College of Arts and Sciences. And I'm still, it's very difficult, but I'm still keeping a research program going, including, as you've heard, undergraduates and graduates. And when people ask me, what's the most satisfying thing about your job right now, it's still the students. I mean, it's watching the students. This commencement, I'll be there presiding as Dean of Arts and Sciences, when Arts and Sciences we actually have 2 separate commencements because we're so large now. But I'll be hooding one of my PhD students who was an undergraduate in my lab. And she came from Columbia, was very unsure of herself. Somebody suggested she goes and does some research in the laboratory, worked in my lab, got very interested, decided to stay on, did a PhD. So I'll be hooding her, and then she'll be doing a postdoc, and she wants to go into academics. So to me, that's the ultimate reward, is seeing, as an academic, is seeing others believe in themselves. And just so you have a little bit of an understanding of my background, I was born in Detroit. My early years where I was born in a public hospital downtown Detroit. No one in my family ever graduated from college. My role models were, my dad was a salesman and my mom was a transcriptionist. My grandfathers were a coal miner and a police officer. And they instilled in me a strong work ethic, but they told me that education is the one thing that nobody can ever take from you. And I see that a lot in Miami. FIU, we're a majority minority institution. So we have over 40,000 students. I think we're now the largest minority institution in the country. I think we have about 15% white non-Hispanic, and the rest is minority. And I love it there. It's very cosmopolitan. I feel an obligation to serve the student body, and so I think mentorship is very important. It's one thing to be a role model. It's another thing to actually work with students and be there side by side with them. So what I'm going to talk about a little bit is some of the experiences I've had throughout my career, and then also some of the research that we've done over the last about 20 years now that I've been at FIU. So I've researched in a variety of areas, but most of my research has gone to the dogs recently. What I do is I study, and I was asked earlier, how did I get involved with this? My training is as an undergraduate degree, I did forensic science, mainly because I was pre-med and they said you have to declare a major. So I flipped through the little catalog and said, oh, forensic science, that sounds interesting, that's my major. And I wound up sticking with it. Then when I was looking to make a little extra money, somebody said, oh, Professor Clausen has a research project doing arson analysis, and you can make a little extra money working in his lab. And I started working in the lab doing that early forms of GCMS for arson analysis, and I said, this is really interesting. I got interested in the research at that level. And then before I graduated, I did an internship. So I went to, at the time, my parents had moved to Orlando, so my high school years were in Orlando, and I But I went back to Detroit and worked in Detroit Police Headquarters Crime Lab, and that's a very valuable experience. In fact, in forensic science, the internship is one of your most important things that you can do, because it's a very small community. If you get good letters of recommendation from people working in a crime lab, that helps you move on into other laboratories across the country and around the world. So when I was there, I got interested in maybe doing more research, and so I stayed there in Detroit, went to Wayne State University. They said, oh, they have a really good analytical chemistry department here. You should go talk to somebody. I basically walked. So anybody who thinks that I had like all this planned out, you're wrong. So I walked over to, literally, walking distance to Wayne State University. I talked to the chromatographer there, Colin Poole. I applied to one university, and luckily I got accepted. They didn't require a GRE, which I was happy because I didn't want to take the standardized exam. And I stayed there, did my PhD, and then I wound up doing a postdoc in the UK doing nuclear chemistry. And I was at FIU while I was doing my tenure-earning years, I was really doing sort of physical chemistry, inorganic chemistry, using separation techniques. And then the director of the crime lab that does the DUI testing, the toxicology lab in Miami-Dade County, got approached by the Miami Herald and said, most money in circulation is reported to be contaminated with cocaine. And is there enough cocaine for dogs to alert? And so I said, well, let's test it. I figured maybe do a 6-month study, put a student on it, and test it out. And we're still doing research on it. I'm going to show you some of it turns out that very little was known about canine olfaction, even human olfaction. In the last 20 years, there's been a huge increase in studying that sense of smell. So what we do in my lab is I started focusing on the A, B, C, D, E, and F of detection. And I'm going to just show you a little bit. We look at the different chemicals and how much is there, how reliable the canines are, how selective they are, and what kind of distractors may be present. Let's see if I can hold this maybe a bit easier. So, give an example. This is a canine working. One of those suitcases contains a bomb. And this is a biological detector that's evolved over hundreds of thousands of years. And you can see the dog just stops, stares at that suitcase until the toy is given. All they're trained is, in order to play, you get a dog that has a ball drive and likes to play with their ball. You say, you're not going to get that ball until you find this odor. It's that simple, and they train them. So I started doing research with instruments trying to figure out a better sensor to replace the dog. The idea, if the dog can do it, we could find an odor, make a machine to replace the dog. And now I'm realizing in my career, we will never replace the dog, and probably not even in the next generation, but we can complement what they do. Now, the big advantage of dogs is that they're intelligent mobile biosensors that can be quickly trained to new odors. So with an instrument, if you want to detect a new odor, like at the airport, when we found out about peroxide bombs being possibly brought onto airplanes in a liquid form, none of the machines could detect peroxide explosives. So you couldn't even just change the hardware or the software. You had to completely change the detectors in order to detect it. With the dog, literally overnight, you could train them on any odor. Now, the number one disadvantage of detector dogs is that they're intelligent mobile biosensors that can be quickly trained to new odors. So they're very smart. Sometimes they're smarter than their handlers, and they can quickly get off track. And so we're doing research and developing best practices in terms of how to improve the performance of biological detectors as well. So you can say there are many, many different kinds of detectors. These are all orthogonal or complementary detectors. I'm going to focus on the ones that are flashing. The work we do in the lab is mostly gas chromatography, mass spectrometry. Many of you are familiar with that. We also do ion mobility spectrometry, which is what they use at the airports. And then with the biosensor, we're mostly dealing with canines right now, but they've trained rats and bees and plants. And I have a project now where we're working on training birds for detection. So the first area is, I'll start with the A, is accelerants. This was the first research I did as an undergraduate that got me interested in research. And I had an undergraduate student work on this while I was a professor, looking at detecting accelerant traces. So there are these electronic sniffers that are sold for doing arson investigations. we found them to be very unreliable. They're very sensitive but not very selective. With canines, what we find is they're not only very sensitive but very selective. And this is an example at the bottom there is Villain, there was a woman who had been doused with lamp oil, lit on fire, and when the police got there, in her dying statement, she said her boyfriend did it and explained what happened. Her boyfriend gave a different account, and Villain here, with his nose, identified 5 different locations in the room. They took the carpeting, sent it to the lab, confirmed that it was lamp oil, and Villain's account matched the dying victim's account, and so he was convicted of first-degree arson and first-degree murder based on the canine and the lab confirming it. Instrumentally, we wouldn't have been able to do that because as I showed you, that sensor would not have been selective enough to locate those particular locations. So most of the technique we use is called solid phase microextractions. Basically, it looks like a hypodermic needle. You see, if you put a material in a vial, the molecules will get into the headspace, the volatile organic compounds. We expose the fiber. Those molecules stick onto that fiber based on the fiber chemistry, and then we can inject it into our GC mass spec. The instrument separates primarily based on molecular weight, in some degree based on size and chemistry, and then you get a chromatogram, the colored chart there. Now they're not really colored anymore, but in the early days of pigment separation was where the chroma came from for chromatography. Each color is a different chemical, the size of the peaks, the amount of that chemical, and then the pattern at the very bottom there is the mass spectrum. Basically, you break the molecule up, that's the molecular fingerprint. So it's the gold standard in forensic labs for doing drug analysis and any kind of identification. We are doing it for the odors, the volatiles above forensic specimens. And it's actually very convenient. They color code the hubs of these speedy fibers based on the different chemistries, so it makes it very easy to select and optimize the fibers. So biotoxins is another area we've worked in. If you look at, these are sometimes called the 3 wicked sisters, 3 of the most toxic indoor molds. Florida, of course, is the mold capital of the world, I think, being so humid. And so it's a big problem when people get mold buildup in their houses. So the 3 you see there are Stachybotrys, Penicillium, and Aspergillus. And these can produce mycotoxins, which can be, trichothene mycotoxins can actually be weaponized and used as biological weapons. I saw one of the posters actually talks about mycotoxins in food out there. And so what we do is the bottom left there is actually essentially a bar graph of each color is a different chemical, and the size of the bar is the amount of chemical. And this the closest thing we've been able to do to try to represent what perhaps the dog is doing in terms of being able to identify an odor. So if you look at those 3 different molds, you can very quickly visually see the difference between those molds, right? Just by the colors. What the dogs do is they do that with their nose, identifying the different chemicals very, very quickly. And the bottom right is a little wheel that they use for training the dogs. And these dogs are just pound puppies. They go to the local pound, they take the dog out, they throw the ball, and if the dog, loves to play with the ball and they just keep wanting to get that ball and bring it back, that's going to be a great dog. Whether it could be a drug dog, bomb dog, mold dog, cadaver dog, or I'm going to show you at the end some human scent work. So since we're on a college campus, I thought I had one beer joke in here, but I always get a lot of grief when I go to conferences. I go to canine conferences, and when I order beer, I like Budweiser, and the cops say that's homicide beer. I said, why is it homicide beer? They said, well, you rarely go to a homicide that you don't find Budweiser laying around. So as I said, I got involved with the idea of, is there enough drugs on money for canines to alert? And of course, a lot of people think that the dogs are trained to, they get addicted to drugs or something, that's how you train them. Nothing's further from the truth. I mean, all they're doing is they're trained to pick a particular odor, differentiate an odor. When they find that odor, they get to play. They've done food reward and other things, but mostly play reward is more often used. Even this year, there was another article in the Mail Online about UK money with every bill in circulation being contaminated with cocaine. So if you go to the airport, you see those ion scans, and you can set them for explosives or drugs. If you set it for drugs, and you put almost any bill in your pocket in the machine, it'll come up positive for cocaine because there's enough cocaine there. So it's really not of any legal significance because everybody's money is contaminated with cocaine. So papers started 23 years ago. The first papers talked about drugs on money. But then that works its way into courts. I'm a forensic scientist, which just means that if the work that I do winds up going to court, that's where the forensics part comes from. Any field can be forensic science. You could be a political scientist studying drug money laundering and be called into court. That makes you a forensic scientist, a political science, forensic science, rather than the natural sciences. So this is where it really started to heat up in '94 when cases where they used dogs to alert the currency as a piece of a forfeiture, say in RICO statutes, they were saying, well, we can't use dogs anymore because there's cocaine on currency. And so what we started to do was research, and our first papers we published actually about 13 years ago showing that the dogs don't alert to cocaine, they alert to an odor molecule of cocaine called methylbenzoate. And so about 9 years ago, the first court cases came out, again showing that canine alerts to currency can be used in forfeiture proceedings as an indication of recent connection with illicit narcotics. Now, I won't go through all of this, but we all know that when you get into research, it's not as simple as you think. So I mentioned, figure I'll do this for about 6 months, maybe a student get a paper, and then we'll move on. Well, there were actually 9 different steps we had to go through to try to figure out whether there was enough narcotics on currency for the dogs to alert so that it would hold up in court, and including exactly how much is there, how much the dogs alert to. Turns out there were volatiles that they're alerting to, how much those volatiles are there, how much of those volatiles could be in non-illicit materials, how quickly do the volatiles dissipate, and so forth. And it sums up in this slide. What we see is that when a properly trained dog alerts, that indicates that that money has recently been in close or actual proximity to a significant amount of narcotics. Then the reason is that they're trained to alert to the odor molecule methylbenzoate, and that molecule dissipates. Okay, so when you touch some crack cocaine rocks down there at the bottom, and then you touch the money, the cocaine molecules transfer the money, but also the methylbenzoate transfers the money. Now, over time, literally within a matter of hours, the methylbenzoate dissipates, and the cocaine stays there. So all money is contaminated with cocaine, but not all money is contaminated with methylbenzoate. And so, this is, I kind of summarized years of work and probably 6 undergrads and 6 graduate students doing research, but it basically means that and this has held up in court, and that's one of the fun things of forensic science is a lot of the research then eventually does get used in court proceedings. It turns out, as I mentioned, dogs can't smell cocaine. It's a local anesthetic. It actually causes it blocks the ion channel, so you get a loss of conduction of signal. So even if you could get cocaine molecules into a dog's nose, you wouldn't be able to train them to alert to it. And this is a very complicated summary of what's currently believed to be the process that's involved in the odorants being transferred across the mucus through transport proteins to receptor proteins and so forth. But the bottom line is you can detect over 10,000 different odors can be identified and detectable. So if you wanted a little bit more simple cartoon, you could do it this way. So if you had like an eye smell, it would hold 10,000 different smells, and you'd be able to differentiate each one of those different particular odors.
I smell, you smell.
Yeah. This doesn't exist yet, but if somebody wants to come up with it, I'm sure the dogs of the world would be very appreciative. And actually, even the dog's nose is very sophisticated. We breathe in and out through the holes in our nose. The dogs actually have this mid-lateral slit, and so it's more of a pumping action. The air tends to come in from towards the front and out through the sides, and it's a very rapid pumping process. They can sniff as much as 8 sniffs every second. At about 30 mils a second per nostril, that's about 3.6 liters a minute of air that flows through the introduction source. As you know, any of the analytical chemists in the room know that sample introduction is always one of the critical steps, and evolution has really mastered it for canines. Now here's a dog's head cut in half. I didn't actually cut this dog's head in half, but when you look at it, there are essentially 3 different major chemoreceptors. They're the trigeminal nerves, and we have the same chemoreceptors. The trigeminal nerves detect spices, so things that are very spicy, that's not your olfactory nerves, those are your trigeminal nerves. So Provost Griffith took a big bite of soup last night. We were at dinner, and his trigeminal nerves got overreacted to the point that he wound up changing his soup out, right? Olfactory nerves are your major sense of what we usually consider sense of smell, where dogs have over 300 million of those cells. We have about 5 million. So dogs tend to be about 50 times to 100 or more times more sensitive because of those numbers of cells. And then the vermonasal organ, or the Jacobson's organ, is for detecting pheromones. Okay, so they use that for detecting their mates. We have a vermonasal organ, but it's not believed to be attached. Actually, in utero it is, and then it's currently, there's some debate over this, but anybody tries to sell you a fragrance that has the pheromone in there, it's not really believed to work as well as it does with most other mammals that have the vermonasal organ. And actually, if you look at those olfactory receptor cells, you'll see canines have many more cells, and relative to the size of their brain, it's a much more important part of their biology. So with the human, you can barely see within those flashing circles the olfactory tract and the olfactory bulb at the base of your brain, whereas the dogs, it really protrudes right out from the brain. But what's really most remarkable about dogs is their sense of selectivity. Okay, we can detect patterns very, very accurately. You can meet somebody once or maybe a couple times, depending on how good you are at faces, and humans can pick out faces very, very quickly. Machines are getting better at it, and they're working on facial recognition of machines. Dogs aren't very good at identifying your face. They use your smell. They use the molecules that you're giving off. And so it's a little bit like if you look at a difficult background, does anybody see what the picture is on the left there? It's a bird in a nest. And how about on the right? Yeah, it's a dog looking down towards the ground. So it's a very noisy background. You can kind of just make it out. If you have a computer try to pick this out, it can't do it, and it's because of that noisy background. Same thing happens with odors. If you take cocaine odor, and I'll show you in a minute, the threshold for humans and dogs is about the same, but if you add, say, coffee odor with the cocaine odor, the dogs have no problem picking out the cocaine odor with the coffee. So sometimes drug dealers will add that in thinking they'll mask the odor. It does nothing for the dog. But what it does is for the DEA agent, they smell the coffee and they check your bag. So it works very well for humans to help them find your bag. So here's a field setup. We just go out basically in these containers. We put these different chemicals that we've identified in the actual forensic specimen and then see whether the dog's alert or not. And this is a slide I was telling you about. The blue triangles are actually the dogs alerting to cocaine base. The red circles are dogs alerting to cocaine hydrochloride with added methylbenzoate. And then the green squares are humans. So these are my undergraduate and graduate students. I tested them to see what their threshold was. And if you look at the dose, this is just a typical dose response curve that you see with any biologic activity. So at about 50% responding is about 1 microgram of methylbenzoate. So I'm not recommending that undergraduate or graduate researchers be used to replace drug dogs, but the point is these drug dogs are not trained to be very sensitive. They're trained to detect large quantities of narcotics. Okay, but they're also very selective. So when you put confounding odors, the humans, when I put coffee in each one of these, none of the humans could even detect any drug odor. So it's an interesting phenomenon. We've done other drugs. This is with ecstasy. It turns out the major odorant is pyperenol that the dogs use to find ecstasy tablets. And then you have to, of course, any forensic application, you have to test non-controlled, over-the-counter materials. Otherwise, you could wind up, putting somebody in jail or inconveniencing them or their civil rights if you were to train a dog on something that's not a controlled material. So we did the same kind of thing, threshold testing. And then what we've been doing is also comparing them to instruments. So this is an itemizer like you'd see at the airport. And so we've actually been able to calibrate these instruments for the odors of drugs and explosives. So rather than just being particle detectors, they're vapor detectors, so they're more like canines. And when we do this, we find the threshold in this particular example the limited detection for the canines was about 1 nanogram, and for the IMS instrument, it was about 2 nanograms. So the dog was a little more sensitive, but they're very comparable. The advantage of the dog is, I showed you in that first slide, they're very mobile, and they can very quickly move. So if you're at an airport, we have a choke point, if I would feel safest if you had a bomb dog just walking along that line. They don't have to be searching. They'll just walk along, and if somebody happens to have drugs hidden in their underwear, like that recent case, dogs are very good at checking crotches, as most of you may or may not know. So a crotch bomber wouldn't get very far with a drug dog or bomb dog. But with the machines, we still have a lot of gaps in our technology with machines. So they had the puffer, and that was designed for particles. You can design it for vapors, which is one of the research areas we've been working on, is trying to make it more of a vapor detector. And now TSA has moved towards these essentially X-ray machines, like the old-fashioned put your glasses on, you can see a person without their clothes. They're moving towards those, which I don't actually think is a particularly good idea. And for the first time in my career, I actually co-wrote an op-ed piece that went to The Hill magazine stating that I think is a mistake to move towards singular technologies like that and get away from detection of chemicals. And then we've also done work with looking at currency and marijuana odor. Of course, marijuana is much more widely used than cocaine or ecstasy or some of these others. And we do find specific odorants for marijuana that we don't find on circulated currency. And then you can also even look at hydroponic marijuana versus normal commercial grade marijuana and so forth. Okay, then another area we've been doing a lot of work in, and it's partly because it's very important, but also there's a lot of money associated with it. In fact, I got a call while I was in the provost's office, and I stepped out, took it, and it was my program manager for a human scent grant I have, and she says, if I gave you more money, could you do some of this with explosives? I go, oh yeah, absolutely. So I'll be trying to round up some more students to work on a new project when I get back. But explosives are more complicated than drugs because there's 6 major different chemical classes of explosives, and then you've got many, many different examples within each chemical class. So theoretically, you could have hundreds of different explosives that you have to train the dog to alert to. So the question is, how many do you use? This is an actual explosive kit from one of the bomb dog teams that we work with. They have 22 different explosive training aids that they try to train the dogs on. And what we've been able to do is by looking at things like smokeless powders, single base and double base, we've identified explosive odorants in those powders, and you can actually replace some of the high explosives with smokeless powders and significantly reduce the number of training aids that are needed. So we're currently doing testing with 6 training odors in a kit in which when the dogs are trained to alert to this, these 6 items, they'll alert to more than 90% of all possible explosives that we know of. So this is an ongoing project that is being worked on right now with a PhD student and 2 undergraduates. And we go out in the field and we basically determine whether the dog doesn't alert, whether they show interest, because usually when you're getting close to a threshold, the dog will investigate but not maybe give their final response, which is, in the case of drugs, maybe scratch, but usually it's a sit. And with explosives, you definitely don't want scratch. You want them to sit down. And then we've done some work with human remains as well. This is one that's very hard to find students that'll work on because it's not exactly the most desirable. So what we do is we look at the various different chemical compounds that are given off of decomposing remains, and then we go out in the field. And so up at the top, you'll see on the left is an experiment where we set up different specific chemicals and then different remains that we put out for the dogs to alert to. And on the right, we have a submerged body in the lake, and then in the background on the right, you'll see they're using side-scanning sonar and some other techniques to try to see what's better. You putting a dog on a boat and just having the dog go around, and basically the dog would just start barking at the water when the gases are coming off of a submerged body. And it turns out it's really best to do both because not always is the vapors or those volatile organic compounds available. Sometimes you may see an anomaly and use an anomaly detector. Other times you won't see anything. The body will be, say, deep down covered with debris, but you get the vapors coming up from the dog. And actually, the little boy in the boat there, if you were wondering why is there a kid in the boat, that's my son. So I had promised my wife that I was, as I called it, babysitting my children. I had twins, a boy and a girl. She said, it's not called babysitting when they're your children. So, but so she went to a conference, and I was watching the kids, and so I flew them to Tennessee. We went to the body farm and submerged bodies and stuff. And my son wasn't all that cool with it, but my daughter, the only thing she was upset about is that we never got to see the bodies in a body bag. And you think things are like really sophisticated on these research projects. The FBI literally got a U-Haul van. We went and picked up a body, submerged it. When we were done, loaded it back in the U-Haul van, took the body back to the body farm, and then they returned the U-Haul truck. So when you're going like moving next time, you wonder what that weird smell is. The FBI might have been doing a research project on that. And then, so of course, what we do is we look at decomposing humans and also decomposing other animals, so beef and pork, and they have pseudocorpses that are available. And what we find is that pork is the closest to a human, and burnt pork actually looks remarkably close to human remains. So think about that when you're having your barbecued pork sandwich. And in fact, that's what they do. When they're training cadaver dogs, they'll train them on buried pigs. And then once they get trained on that, then they'll proof them off of pigs and train them on human remains, because humans and pig tissue is very similar. There's a lot of biological research done in that area, and we see the same thing in terms of human remains. So I won't get into all the different chemicals that are associated with it, but they're mostly, we believe, are disulfides and then human scent compounds, some of the butanoic, pentanoic, hexanoic acid, and so forth. And then lastly, I'm going to just talk a little bit about human scent. This is what most of my research has been focused on in the last 5 years. I got approached by the Netherlands National Police that they were being challenged in court over whether human scent is unique. And they do human scent lineups across Europe. They find a crime scene object. I mean, they have a suspect. They'll take an object from a crime scene. They'll try to identify a match of the odor from somebody who might have touched an object to a suspect. And they do essentially, like eyewitness lineups with humans, they do scent lineups with dogs. And it's held up in court, except for there was a foundational issue of whether human scent is unique. It had never really been formalized. So what we're looking for are primary odor chemicals that are stable over time. They don't vary with your diet or with environmental factors. And what we try to do is isolate out chemicals that we know to be secondary chemicals that come from your diet, and tertiary chemicals that come from outside sources like the soaps and fragrances and perfumes and so forth. And this is a Schlieren image taken by Gary Settles at Penn State lab. And since your body temperature is typically higher than the room temperature, you have a human thermal plume, almost like a chimney. So you have a constant heat, a warm air rising and going up and over your body. And on that are carried hundreds of skin cells every second that your body sheds off or coming off of your body. So wherever you walk, you leave a trail of your skin cells. And so your body contains...
Don't move.
Yeah. So you have about 60 trillion skin cells, and it's estimated that about 50 million cells die every second. And of those, maybe each skin cell that's coming off your skin has been there for about 36 hours, so it's saturated with your odor. And you're shedding about 667 cells every single second. So it's believed that that's what the dogs are following to figure out where a person's been. And the longer you've been somewhere, of course, the more of your scent you leave. And many of you may or may not be aware, but the human skin is actually the largest organ of the human body. It covers about 25 square feet, and it represents about 10% of your body weight. So this is an image of from Body Worlds of a person holding their skin to give you an idea of just how much is there. Of course, that's exposed to the environment and is used as an emerging forensic specimen is human scent. And so there's 3 different ways that it's used. Sometimes it's for crime scene response. Where does a trail lead you? If there's somebody There's specialized teams the FBI has where a case goes cold after 24, 48 hours. They'll bring in a specialized team where dogs are trained to follow aged scents that are a day or 2 or 3 days old, try to go to a location. They may do it for suspect identification to match, as I said, an object that they find that was used at a crime to a person, or they use it for location checking to set up for surveillance. So it's presented different ways in different countries. In places like Finland, they do a tube retrieval system. So as I mentioned, it's like instead of a 6-pack of images for a visual lineup, they do a 6-pack. Number 7 is actually a control run of scented bars, and they see whether an object that's presented to the dog matches one of the bars. In Poland, they do it with cloth responding system, they call it. So they basically use a cotton material, and they do the same thing where the dogs are run with these cotton materials. And in the US, we use what's called a scent transfer unit, which basically looks like a dustbuster. I think that's what it is, really, just with a hood on it. And then now there's been research done looking at various different kinds of what are called human scent collection units. Basically, they're just vacuum systems for collecting scent through sorbent material. And I'll show you how this is run. This is a human scent lineup here in the US. Very rarely done, but this is one team that I investigated out in California where they just had boxes and then the dog would sit down when there was a matching scent available. In Europe, it's a little bit more sophisticated where they take these clean stainless steel bars, so the suspect and then other people who are the same age and race and sex, similar to what you do with the eyewitness identification, touch bars. They'll put it in a lineup. This is a lineup in Finland where I went as part of a research project. And I brought actually both a grad and undergrad student out to a conference there in Finland. And you'll see when the dog matches the scent, that's its primary reward. So the dog likes to play with those stainless steel bars. Okay, this is a Belgian Malinois, so they're like German Shepherds on crack. I mean, they're very, very, wiry, wild dog. And so they literally just love to play with those bars. And they only get to play with those bars when they get a match, And if they get the wrong bar when they're doing control runs, the bar just stays there, and then eventually they know they don't get their toy and they have to move on, and that's how they do their training. Now in the U.S. we do bloodhounds. So this is a bloodhound where there's a negative trail. So they're trained that if there's scent from an object is given to them, doesn't match the scent on the ground, the dog just sits there, or this dog is actually vocal. If there's a match, then the dog takes off and follows the trail. And some of these trails can go literally for miles, and they can be aged for several days. And most people, including myself, when I first got involved with this, I wasn't really believing it. It seems like a lot of folklore and not a lot of science behind it. So we've been doing a lot of work on doing testing of these teams. So this is what the scent transfer unit, like I said, just basically looks like a dustbuster. It was actually featured in one of the CSIs. I'm not sure why he was holding it there over a dead body, but because as you know, most of the stuff in CSI is not real. My mom will call me up, "Hey, are you watching CSI?" I go, "Mom, I don't watch CSI. It drives me crazy, that show." But, so we're looking at a variety of different areas. So we've got students working on all these different areas: stability, dissipation, the collection material, contaminant storage. Even within the collection material, we're looking at various different kinds of coatings on materials, or just if you use materials that have polyester or rayon, wool, cotton, and so forth. The idea is to come up with a simple material that can be law enforcement can use that might be better than what's currently used. Most of the time, it's just a cotton material. And we're also looking at flow rates. So you'll see that the same individual with different flow rates, it can make a big difference on what the odor looks like. This is just using a PCA analysis. And then we look at contact versus non-contact. So contact sampling, we find you collect about 10 times more odor than if you don't contact the samples, if you do a remote sampling. The problem with contacting it is you disturb it. So if there's DNA present, you can disturb it, or you can cause other problems with it. So ideally, you want to do non-contact. And there are also individuals who don't have business cards that are interested in doing non-contact sampling of people, as you can imagine. If you can find a unique odor of a person and not know that you're taking their odor, and keep a database of that. There's an interest in that kind of work as well. So again, visually, here's just to give you an idea, 5 females at the top and 5 males at the bottom. And if you look at those, if I give you just a second to look at them, you can see that they all look different. You know, there's no 2 people alike. And of course, we haven't done every single person in the world to say that things are unique, but we do see enough, and these are the primary odor chemicals. You actually see hundreds of compounds when you look at just sampling a person and the VOCs that are given off by a person. These are the compounds that are reproducible over time, and we've eliminated the things that we know come from fragrances and from things that the foods that you eat and so forth. And so, of course, this would be pretty difficult to just do this visually. So when we do statistical techniques, we're using a technique called spearmint rank correlation. Basically, the most abundant compound gets a 1, the next most abundant gets a 2, and so forth. You can actually run a statistical technique and use the threshold of, say, 80 or 90% correlation, and you can correlate all of the sampling from the same person to themselves and differentiate people who are not, that are different individuals, even if they have similar age and race and sex and so forth. And so the ultimate test would be identical twins. So this were 2 undergraduate students that happen to be identical twins living in the same house, eating the same food, more or less. They actually shared a room and everything. And you could still differentiate them. They only differed by 2 chemical compounds. And so there's about 80% similarity for these 2 identical twin boys. Versus if you take fraternal twins, we had 2 other students that were fraternal twins, and there was only about 55% similarity in their odor constituents, which is pretty interesting because it comes pretty close to the genetic makeup that you'd expect being expressed as these VOCs. So as I mentioned, it's interesting doing forensic work that you get to go to court. So back in 2005 was the first time this was challenged in what's called a Fry hearing. Kelly Fry is a standard that California uses and some states still use, where you need to show general acceptance in the scientific community to use it in court. In this case, the judge did allow the use of the scent transfer unit and the idea that human scent has sufficient variability to be used to differentiate individuals as long as the proper techniques were used and so forth. And the most recent case was just decided last year. This was a case in Alaska. There was a homicide, a serial killer. His last victim was found in Wasilla, Alaska. We all know who came from Wasilla. This was not connected with Sarah Palin, but
but it was close to Russia, right?
Yeah, well, I was there and I couldn't see Russia. And so in this case, what happened was this was a serial killer who had been arrested previously. He had gotten the evidence, or the defense attorney got the evidence suppressed, and so he was acquitted, and then he wound up killing again. So the feds came in, and they brought in a whole team, including these bloodhounds that were specially trained, and they trailed from an ATM machine where there was an image of a hooded person taking money out of the victim's account. They trailed miles from that ATM machine back to a house, and they got a search warrant, searched the house, and they found evidence, DNA evidence and others linking him to the murder. So I actually spent an entire week out in Alaska. I even went mushing with the dogs team. That was kind of fun. And it was pretty interesting because at first the defense was pushing very hard against using the canines and human scent uniqueness and so forth. And in the end, I can call him the serial killer now rather than the suspect, because rather than, since the feds came in and he had committed murders in multiple states, they went for the death penalty. So to avoid the death penalty, he confessed, and he remarked how remarkable the dogs were and how accurate the dogs were. And so he wanted, I don't know why, but he wanted to be in the state pen rather than the federal pen. So I think he got like 30 years in state pen, and if he's still alive, then he spent the rest of his life in federal prison for his acts. So it's kind of rewarding. I guess the second most rewarding thing, other than working with mentoring students, is sort of folklore and just practices that were done by certain groups and not so much necessarily best practices. And so this is started back in 2004. It's been funded by the FBI, the National Institute of Justice, the Department of Homeland Security, and the Technical Support Working Group, which is an interagency counterterrorism group, basically setting up these best practices. So I won't go through this, but there's a whole 10 different subcommittees that are working on this. And essentially what we're doing is looking at, again, what's the best method for detection? The canine for vapor detection, maybe a particle detector like an instrument. Ultimately, your GCMS in the laboratory where you can look at vapors and particles, the best detector available, but it's not very robust. The miniaturized GCMSs just aren't there yet. And of course, as I mentioned, the ideal is to have a combination, a layered approach. So as I mentioned, also, this is what the TSA is currently doing. I went through one of these puffers yesterday coming up here to New York. I wasn't very, worried about it, even if I was a terrorist. I know how to get around actually not a puffer machine, a X-ray scanner. So they eliminated the puffer machine and they put in one of these. So basically it just looks at any objects that you have on your body. So of course if you have an object concealed, you can use your imagination how you can conceal an object, because you don't need a lot of explosive to take a plane down. The anomaly detector is not going to pick it up, so you need a vapor detector as well. Otherwise, this is even easier to thwart than the previous instrument that it replaced. And then the very last thing I want to mention, we have a paper coming out in Forensic Science International next month, where we've actually taken, this is a pickup truck in which we put a fairly large amount of peroxide explosives here in the passenger side. We've had a person touch the steering wheel and act as the driver, and then a terrorist touch trigger device in the peroxide bomb. It's a little hard to see here, but the vehicle is completely destroyed. Then we collected objects from the vehicle, pieces of the steering wheel and so forth, used the scent transfer unit, non-contact sampling, presented it to the canine teams. This is out in a desert region. The dogs were able to trail into the desert, into a village, and identify the bomber from scent after detonation, which we did a double-blind scientific study. Everything was videotaped every step of the way, and it's taken a little while to be allowed to be published, but it's now going to come out, and it's pretty remarkable. This gives you an idea from the blast site. The dogs, there was essentially a Y split, and the dogs could go out into the village, and they were able to identify the specific person that had touched the object. And actually the person or persons that we used were from my office, my research office, and my students that had never been there before. So these dogs had never experienced these people before. So overall, just hopefully I gave you some examples of cases where canines are still the most reliable detectors available, but they do have variable performance. So we're trying to have these best practices implemented to improve their reliability. They can be great models for instrumental detectors, but ultimately they really should be used in a layered approach. We should use biological and instrumental detectors. And we're developing calibrants to allow improved performance of both canines and instruments out in the field. And as the literature it improves, the performance will improve, as will the courtroom admissibility. And a case in point is human scent differentiation, which is demonstrated by canines and supported by these instrumental methods. And every one of these techniques I showed you did involve both graduate and undergraduate researchers. So I tend to try to, as dean, I don't have a lot of time, so I have meetings with my whole group, and then on an as-needed basis I meet individually. And so I tend to have, partner up undergraduates with PhD students, and then usually at least one postdoc or lab manager to kind of help manage things, because I don't even know the password of my lab anymore in terms of my computers and stuff. But I also find that that's useful because the undergraduates get to see graduate students doing work and moving on. And so in the next month of analytical methods, I think it's the first time in the history of this Royal Society of Chemistry journal there's going to be a dog on the cover. So Paola Prada, who's the first author on this paper, she was that undergraduate that I told you about. She came from Columbia. Actually, my wife, who's a school teacher, had her in high school, and she said, you know, wasn't sure what she wanted to do. She may be interested in chemistry, and she was thinking about FIU. And so my wife said, oh, I'm sending this person to talk to you. And I'm like, don't send me people. I don't have time to be, you know, talking to somebody who doesn't know what they want to do or whatever. Anyway, you can't argue with your wife. So I wound up talking to Paola and said, well, why don't you come do a little work in the lab and see what she thinks. So she started as a freshman working in the lab and said, I'll be hooding her next week at commencement with her doctorate. And she secured a postdoc funded by the CIA. They have a postdoc program. So it's a non-classified project, but it's something of interest to the intelligence community. And she wants to be an academic. So, you know, it's one of those things that, again, it's extremely rewarding to see the, you know, that light turn on. And that's really, I believe the reason I'm an academic is because I went to go work in Dr. Clausen's research lab doing that arson project as an undergrad and got that first interest in solving a problem. And then, of course, when you get your name on a poster and maybe you get your name on a paper or stuff, you realize that's still exciting. Whenever a paper comes out, I get so excited to see my name, just like when the first one, maybe over, you know, I don't know, it could be hundreds, and I'll still get just as excited every time. So Chris Clausen's the reason I'm a scientist, and Ivolov Griffiths is the reason I'm an administrator. So you want to. So I want to thank, of course, I didn't do any of this work. It's all my undergraduate, graduate students. So the underlying names are my current undergraduate students that are working in the lab. And of course, the people who fund the project. So that's not actually my research group. This is, so, you know, playing rock bands. You know, so I may be getting up there in years, but use my kids' rock band set when the group comes over. And again, thank you for your inviting us. And,
Let's have another round of applause for Dean Ken Furton. Looking at that picture, I just think every dog has its day. Just, not to hound you, but I wanted to ask you for your recommendations on body soap. Perhaps you have some suggestions, because it could be very uncomfortable in the future when you walk through JFK and they say, sir, can you please raise your arms, and we'd like to sniff you a little closer. So you can imagine what the unimaginable was coming to light, but science will bring us there. So before I continue with other bad jokes, I'd like to invite Louise Miller to direct us to, we want to do some questions. Okay, do we have some questions for Dr. Furton? There is a question. Okay, Odingo, go ahead.
My question is, how will this technology work if somebody actually gets into the car and drive away?
like for following scent in a car?
Right.
Yeah, that's where, that's a great question, because that's really where some people are say they're following car trails, but it hasn't been confirmed scientifically that you can do that. Anecdotally, the idea is if you're using your own car that you use on a regular basis, that your car's not airtight, even if you use that, recirculating thing. So there is odor coming out of the car, and they've shown that dogs can follow car trails, but only for limited distances, and really if it's your car. Because if you go, now, are there any felons or future felons here? Because I'm giving away trade secrets. But if you go and like rent a car and then you're driving in it, there's not enough odor coming out of the car for a dog to follow. But it's a little bit controversial, because what happens is you'll test dog teams and they won't be able to do it, and it turns out they're not trained to do that. So dogs are only able to do what you train them to do. So that's an emerging area that we know that people leave a scene, and then if they get into a car, you need to really set up scenarios to train dogs to follow car trails. And the amount is extremely low, so you're bringing the level down even lower. it's also a challenge because instrumentally we can't confirm that. We can do, I can do contact sampling, and I can confirm in the lab that this is this person's scent, and I can match it to them and so forth. But when you get to like that exploded debris, after somebody touches a bomb, you detonate it. And it's been shown, they've used this in theater, in Iraq and Afghanistan, that have a roadside bomb, the dog can actually trail and locate potential bombers. Well, we can't identify, we can't do anything with the material in the lab. The level is just too low. The same thing with car trails. It's just the amount is at least below current technology. Maybe down the road we'll be able to detect them.
Hello. I have a question. This whole research, like, I feel like is there any mistake that dogs could make a wrong assessment on a suspect?
Absolutely, yeah.
How does that happen?
Well, what happens is that's one of the, even though we've been able to show that you can take identical twins and you can differentiate them based on their scent, the reliability of dogs to follow a specific person's scent is about the same or maybe a little better than eyewitness identification. And we know eyewitness identification is fraught with potential problems because it depends on how long you've seen the person for, what the lighting conditions were and so forth. And so it's, I always have to be very cautious to say that this is not like DNA matching. We have a project right now where we're looking at, where we sample somebody, let's say we have a scented sample. In theory, if you do low copy DNA, you might be able to get DNA off of there to identify a person. So this is not identity of the person. All it means is there's a scent association, and you have to be very careful to let the jury understand and the judge know that it could be somebody else. Just like eyewitness identification, it depends on, on the conditions involved and so forth, and people get misidentified regularly with eyewitness, and you can misidentify people. At least what tends to happen, at least, is most often when the dog doesn't get enough scent or doesn't get a matching scent, they just won't follow a trail. But what we're looking at is, anecdotally, there's a thought that depending on, say, your biochemistry, the dog might misidentify people, say, if you're a diabetic. So we have a study right now where we have a healthy population, we have a population of diabetics, and a population of people who are clinically depressed, trying to see whether there might be a similarity of the odor and you might misidentify. And this comes from talking to investigators who said they're looking for a violent sexual predator, and they have a scent sample and from a crime scene, and the dog follows a trail and it goes to somebody's house. What they'll typically do is rather than that's may not be enough to do probable cause to search the house, they'll just do set up a surveillance. And then somebody's acting unusual, or they give a reason that you could get a search warrant to check. And when they go in and they find out, they sometimes misidentified a person, but it turns out that that person's also a violent sexual predator. But they're not associated with this crime. So that means maybe there are similarities, and you can, which we don't know at this point. So it has to be done very cautiously. This is not DNA fingerprinting where if a dog matches, that does not mean that that's that person. It just means, and in fact, what it's used more often than not, same thing with DNA fingerprinting, mostly it excludes people. So they have a lot of suspects, and you can exclude most of the suspects because there is no scent association. But when you get a scent association, you can't ever use that by itself. Same thing if the dog alerts to currency, you can't take somebody's money because the dog alerted to currency. But what that means is the person has to explain, why is there drug odor on your money? And I'll give you an example. We were working a research project at Miami International Airport. We had put up containers, and the dogs were running, looking at those thresholds. And then the police officers get bored, so they start searching near people getting off of planes and near the luggages, right? The luggage carousels. And so the dog alerted to this guy's pocket. And so he goes, "Sir, can you empty your pocket?" And he emptied his pocket. He had a huge wad of cash, and he had a one-way ticket paid with using cash. Those are key signs of somebody who's doing something that he's not supposed to be doing, right? So the dog alerted, that means drug odor. He's got cash. He's got a one-way ticket. And so you get a dog alert, and then usually you get a stupid story. Like, oh, well, I'm waiting for Fred, and Fred never shows or something. Well, this guy said, well, I'm actually on a conference, and that's not my wife, okay? And we were smoking marijuana in the car, you know, on the way to the airport. So after laughing a while, they let him go because he had a reasonable story on why he was carrying cash. He had a one-way ticket. He had odor on his money, so that's not enough to take his money. But if he had come up with a different story, then they could have initiated a civil forfeiture proceeding to say that that was indicating that it was the proceeds of illicit activity, and then he'd have to go to court and explain that. So you can never use any one piece of evidence like this. Any case that hinges on one piece of evidence, even if it's DNA, is always very suspect in my mind. So, good question.
I've heard things about odors or chemical signatures being used for medical diagnosis. For example, certain types of cancer can be detected by dogs. Do you think that this is a field that should be pursued?
Yeah, absolutely. It's interesting because they're doing volatile organic compound detection, like say blowing into a machine like a breathalyzer, has been shown. There are compounds for like lung cancers and things that have been reliably proven that can that has potential for office visits and so forth. But in the canine detection of cancers, there are conflicting stories. There are some studies showing that it's not reliable, and that maybe the dogs are detecting basically when you have cancer, you have breakdown of human cells, and it's almost like a cadaver dog kind of thing. So the reliability has not been proven with canines. I think it's probably unlikely that there's going to be a lot of application of canines sniffing. I mean, if you're worried about the dog sniffing your armpit, you imagine if you're going in there and you want to get checked for cancer and, you know, prostate cancer or something like that. I don't think I think I'd rather have a machine do that in that case. But it's definitely worth pursuing because, like I say, biological detectors, they've evolved and they know things that it'll take us maybe 100 years to develop on our own. So it's definitely something that should be pursued.
I just had a question about, you have dogs that sense nuclear material for like radiological bombs or whatever?
I could tell you about that, but then I'd have to kill you. So, no, but there are dogs trained for contraband dogs, they call them. So everything you can imagine from, let's say, bugs in a room. I'm not talking like termites, but usually it's not the nuclear material, but it would be things that are associated with nuclear materials. If you think of, you know, electronics that go into trigger devices, things like that. Dogs, because they're so mobile, and you can train them to anything, you can just use your imagination. If anything you want to try to recover, the best technology we have right now is a dog. The problem is that, one of the problems is that dogs are pretty obvious. So you can't, like, you know, just release one of those crazy Belgian Malinois in somebody's office and search for a bug in the room or something. So there are also breeding programs looking at how to make dogs look like mutts, but they're really more like a purebred dog that are very reliable. And this is for military applications as well, because the first thing that happened when they started deploying dogs out into the field to take a well-trained, beautiful-looking Malinois that's out there, all the terrorists do is just shoot the dog. So what you got to do is you have to make a dog that looks like a mutt that's actually patrolling. So there's a lot of subtleties that are associated with that. But yeah, so dogs are great models, I think, for figuring out ways that you could develop better sensors. But also we've only scratched the surface in terms of what you can train a dog to alert to reliably, and how do you make that deployable? Because one question comes up is, are some dogs better than others, like breeds better than others? And you see more variation within one breed than you see between breeds. So people select dogs usually for physique. So, you know, police like to have a big, intimidating dog, you know, their shepherd or their Rottweiler or something like that. It's not because their nose is particularly that sensitive, because you can, I mean, my pug, I have 2 labs and a pug. My pug's got a great sense of smell, but it's not very intimidating. I mean, he can't even pick up a pencil. But you could train him. I'm sure you could train him for drug work. I mean, one of my labs is a drug dog. I forget what better way to keep your teenagers clean than to have a drug dog in the house, you know. But,
One more question?
You said that CSI is not real, and I know a lot of high school students who would say they want to go into forensics because they watch CSI. So what do you do when they come to college and they want to do it, but it's not what they're thinking it is?
yeah, that's a great question, because the whole CSI effect, I mean, I'm old enough that I watched Quincy. That's how I got interested in forensic science, and that wasn't real either. I mean, but because I realized they would be saying, oh, the gas chromatograph, and it was really an infrared spectrometer or whatever. But that wasn't quite as obvious as CSI is, like where they, and my daughter is, I could never convince her to be interested in science from my doing demos and stuff, even from elementary and middle school. But she now wants to be a forensic scientist because of CSI. But now she's realizing, okay, they don't get to drive around in yellow Hummers and shoot people and then bring the stuff back and all that. So there are aspects of it that are real, but you have to understand that one person on CSI is actually like 4 people in the forensic community. And I think it's important that students understand that it's actually much broader than what you see on CSI. I mean, there are people that do forensic accounting and all sorts of merging areas within forensic science. So I try to keep them interested and find out areas they're interested. They might want to do anthropology, or they might want to do some other area. Because CSI has been great for stimulating interest in science. It's considered, forensic science is considered a gateway discipline, especially for women in science. We have a pretty big forensic science program. We have over 100 students in the undergrad and grad program, and they're almost all women. We don't see that. You see more, increasingly in chemistry, you see more women, but nothing like that where it's almost all females, and certainly not still in physics and some of the other areas. So I think it's been great to kind of get you know, an attitude change. I can tell you on the legal side, the CSI effect is incredible because juries are expecting a lot more. So if you go into court with your old-fashioned, you know, stuff, which could be perfectly valid scientifically, they just watch CSI and they're like, well, wait a minute, I didn't see you, you know, do scent transfer unit and run this and do all that. And so it's getting harder to get convictions in court because of this, what's called the CSI effect. So it's helped bring, I think, students into the scientific areas, and they just need to understand that it's not as exciting as that, but it's still pretty darn exciting in my mind. I mean, it's depending on what you find interesting, but it is a problem because a lot of students will come. What we do at FIU is we're looking at doing like a forensic. We have mostly forensic science, do like a forensic studies program where we do intelligence studies and we do forensic psychology and some other areas, so that when students say, well, I guess I'm not going to be a forensic chemist or a molecular biologist, but maybe I can do psychology, or maybe I'll do, I'm good at languages and I'll do forensic investigations for intelligence purposes in the intelligence community. So there's a lot of different sort of areas that it can feed into. But that's a great point, because you know, there are many more students interested in it, but most of the jobs, at least that people immediately think about, are criminalistics, which is crime lab, which is mostly biological and chemical techniques. And so if chemistry and biology is not your forte, I mean, my wife, she said she never worked as hard as she worked to get a D in chemistry when she was at Purdue University. So, you know, she just could never imagine doing, you know, this forensics in that regard.
I'll just use my classroom voice. Let's give Dean Furton another round of applause. Ken, we have a practice of not allowing our guests come and leave without something. And I know that hurricane season is coming up in Florida. So I want to equip you appropriately for hurricane season with a York College polo shirt. You can run without And then you can put this on when it's raining a little bit. And this is for Debbie.
Great.
Thank you very much.
Thank you. Participated in those and how we can do more for next year and do better. Now I'd like to invite Kimon Morris and Nicholas Hunt Walker to come up and join us in a panel. They're going to say a few words each about the experiences of, come, you guys can come forward now. As you're walking up, I'm going to be continuing to talk. And they're going to talk to us a little bit about the experiences of being a student at York, engaging in undergraduate research in one form or the other, and how it has affected their academic career. Both of them are on their way to or currently in graduate school. So let's give them a round of applause. And let me invite Nicholas to begin by maybe telling us a little bit about his experiences with undergraduate research and how it formed the outlook for his future plans, and then he can tell us about his graduate plans as well. No.
Hello? Okay. So I've been pretty lucky because I've been able to connect with a large network of people, and so.
Do you want to talk firstly of nature?
Oh yeah, I was going to. That was the next sentence. So I'm currently a graduated senior in the physics and Earth and Physical Sciences department. My major is physics and mathematics. My intent was to go into astronomy. When I started my undergraduate career, that wasn't my original direction. I was actually going to accounting, and then I took an astronomy class and I loved it. And since then, I've been able to move through a network of people, and in that time, amass a large amount of research experiences, both at York College and at other institutions like the American Museum of Natural History, Columbia University, and the University of Wisconsin. So, do you want me to be specific, or?
Well, you want to maybe talk about one of those research experiences that was extremely formative and your relationship with your mentor?
Yes. So I would say that my most influential research experience took place in the summer of 2008 when I had my first real research experience at the Museum of Natural History. My advisor was a professor at the College of Staten Island, Charles Liu. I'd known him before because I had taken a class at the museum, Introduction to Space Science, and he had liked me as one of his students. He wanted me to come back to do research the following summer. And it was my most influential, not necessarily in a good way, but because it taught me to be an independent researcher. And I didn't know this. I didn't know anything about research when I first started doing physics. So I didn't realize that most of the time you'll be left on your own to do your work. I mean, now as an undergraduate, oftentimes your mentor will be there by your side through every step of the way. But as you get more serious into it, and as you go on into graduate school, you're increasingly left on your own. And so he kind of threw me in the deep end, and I was able to emerge successfully, I think.
And you've been accepted to several graduate programs in physics.
Yeah,
that's
in astronomy.
Yeah, in astronomy, which is, you want to tell us a little bit about that? It's very exciting.
About the application experience?
Well, first of all, what you want to tell us what you have, where you've been accepted to PhD programs, and secondly, maybe talk about how your undergraduate experience in research was a key factor in your applications.
Okay. So I applied this past fall to 4 institutions, the universities of Washington, Wisconsin, and Arizona, and also Columbia University. These are all some of the top programs in the country in astronomy. Particularly the public universities are the top, like, non-Ivy League schools for astronomy in the country. I was accepted to all 4 schools. And I would attribute that acceptance to the very fact that I was able to have so many research opportunities. Because when you go to the graduate level in astronomy, you know, everyone's an A student, everyone's good. So what they rely on to make the decision, besides like GRE scores and your academic record, is letters of recommendation from research advisors and your research experiences, and also the quality of that experience and how well you know what you did. Because you can just say, you know, I did this. But if you can't explain it, then that means you didn't really get into it. And if you didn't get into it, that means you're not a good researcher. And they want someone who's ready to go. They don't want to have to train you from the ground up. So I would say that without the research experiences that I've had here in the physics department and away, I would not have been able to make it where I was able to make it.
So your experiences in research, it sounds like, came both in York and outside of York.
Yes.
Can you talk a little bit about the particular experience of being a student at a small undergraduate college like York and how research in this environment worked for you?
So on the student side, the non-research part, I was able to benefit from the small class sizes, especially in physics, where we only had about six majors at one time in class. We had very small class sizes, so it was very personal, and we were able to benefit from that in being able to relate to the professor on a very individual level. On the research side, being here, I was able to. I mean, it wasn't much of a hassle to leave the campus to have to go somewhere else to do my work. I would just, you know, I'd get out of class and then I'd go to the lab, and I'd do work. And then when class time came again, I would, you know, leave the lab and go to class and then go back to the lab after that. So the accessibility was great in both of the research experiences that I had here, both in astronomy and in nuclear magnetic resonance. So I think that being here, it offered a unique ability.
Great. So you'd recommend other students who have ambitious career plans to definitely try to have that experience?
I would say without a doubt that you should get into research as soon as possible.
Okay.
Seriously.
Okay,
because the more experience, I cannot stress enough how much experience counts. The more experience you get, the better you're looked upon. So if you know of a professor who's looking for undergraduates to work in their lab, you should go and find them and work.
Thank you. So I'd like to now move to Ms. Morris. Let's thank Nick for his comments. Thank you. And we move now to the School of Business and Aviation Management, which, Information Systems Management. The aviation program is under there. And I think we wanted to have a diversity of experiences on the panel. Ms. Morris, just to give her a brief introduction, is the lead on the VITA program here, working closely with Dr. Robert Clovey, Professor Clovey, and she is now, well, I'll let her tell us where she's going to graduate school, but her experience with undergraduate research came in the form of hands-on experience in managing a very, very large accounting program designed to help low-income families. So let's invite Ms. Morris to have a few words.
Thank you. My name is Kimon Morris Haynes, and I was a class of 2008 graduate student here at York College, where I did my BS in accounting. I am currently undertaking my master's in taxation at the Zicklin School of Business at Baruch College. My application process was very simple. I only applied to Baruch because that was the only place I thought was fitting for where I wanted to go. So did it one shot, got in, and I must say everything came from my experience with the VITA site, as I had no other accounting experience, period. When you are actually going out there to apply for these colleges or to even apply for jobs, you will find that they tell you it's either an education or experience, and they want both. And how is it that you get experience while you're pursuing the education? Oftentimes, it's very hard to do that. So I was blessed in that after I finished college, I was able to be involved with the VITA program, and that is where I ended up getting my experience. The VITA program pretty much is a IRS-sponsored program that's been around for over going on 41 years now. We have maybe 40,000 VITA sites throughout the U.S., and we pretty much do free tax preparation for low-income tax individuals. My research type-based experience is very different from what you would find with a chemistry major, in that you might know exactly what you are researching at that time. For me, it's very hands-on. Any specific day, a taxpayer might come in with this scenario, and I just have to know where I'm going to get that information and how I will present it to them. For instance, early out in 2008, when we were doing 2009, when we were doing 2008 returns, we found that a lot of our international students were getting their returns prepared as if they were regular residents of the country, and no one at the site at the time had any type of experience on how to deal with this situation. So I went on the IRS website, got all their publications for that, and pretty much tackled that scenario. This specific taxpayer had done 3 tax returns that were done as a regular 1040, and it's April 15th, so I guess it's fitting to be talking about taxes. She did 3 tax returns that were done on the US 1040, which, if you're an international student or scholar or teacher, you must do your return on a 1040 NR. So you're looking at credits that she was not entitled to, were all given to her in previous tax years, and once the IRS gives you a refund that you're not entitled to, they look at it as a loan that you now have to pay with interest since April 15 has passed along. So I had to go in there and really, really sort out all of her documents. It took me about 3 weeks to put everything together. Finally, when I was able to, after I found out all the laws governing international students, she had to pay back very small amount, even though she had gotten thousands in refunds that she was not allowed to. She was only had to pay back $160, and the government, by other taxes that they were not supposed to be taken from her checks, they would have to come back and pay her $600. So we were able to help this taxpayer out. And as I said, it is very interesting, and every single day it's a learning experience, and the tax law changes all the time. And I must say that I went into accounting thinking, okay, I'm going to be dealing with numbers, simple plus and multiplication, a little percentage, and that's it. But I was very, very much deceived, because accounting is not only about numbers. It's very big on communication. You have to know how to write well, speak well, and it's also, there's a lot of research involved. If you go in the field of auditing, all you're doing is research. You are researching documents. Yes, numbers do flow in, but that is not the core or the gist of it. So for any accounting majors here that think that you're only dealing with numbers, you have been fooled. It's not numbers, it's a lot of work, and you have to know how to go out there and network, meet with the right agencies, and take all the necessary steps to not just come into college and take your classes and leave. You must realize that college is all about networking. It's all about gaining that experience. And even though you might be on campus all the time and you cannot get the experience in your field, you have to find out the outlets that's available to you on campus that can be seen as experience. And as I do believe that my time with the VITA program, and if you're an accounting major or even not an accounting major, the tax law affects everyone, and you should check it out, come be a part of it. It's a great, great, great learning experience.
Wonderful, thank you. It's really a unique kind of experience. Let me follow up with a question to you. Well, first of all, so the program at Baruch that you've been admitted to, what is the typical amount of experience that they would require of someone that they would accept?
Well, first off, Baruch MS in Taxation program only elects maybe 50 to 60 students per registration period. So it's very competitive, and they normally look at a year to 3 years experience based on if you're going to be specialized doing an MS versus an MBA. And so I had, as I said, no experience at all separate from my VITA experience.
So this program essentially was able to give you that additional piece.
Yes, it definitely did, especially after I came out of the GMAT exam and I was not so not so happy with my score. But on my essay, I did put that, I do not think I should be judged by a standardized exam, especially with all that I have to offer. So I did put that out there.
Could you speak a little bit about the experience of being, the same question that I asked Nick, of being at York? It's a small campus. We're focused on undergraduate education. We do some things particularly, and we do it in a certain way. How did this particular environment amplify or connect with the experience that you had?
Okay. Well, first off, with York colleges, being an accounting major, it's kind of frustrating because there's not really that much outlet for you to either get the type of internship that I've seen other majors or the experience that other majors are able to get. So I found it that I would be the student that came into class and I was out. It was just in and out, and I never really sticked around. It wasn't until the FIDA program came about at York College in 2008, so it wasn't until my last semester, pretty much, that I did and was able to get this type of experience, and it's been great. We have rotating volunteers of probably, when we started, maybe 25 students, to now we probably have, on a rotating basis, maybe 250 students spanning 3 different CUNY colleges that do volunteer with our program. So it has grown, and it's a great opportunity for everyone interested.
Wonderful, thank you so much. So we have time for a few questions. I was hoping that some of the students might have some. Are there any questions for either of our panelists about graduate school, research, anything in that area? Yes, question here. There's a mic coming to you right now. Go ahead.
Is there any reason why you chose a CUNY college for your grad school?
For my grad school, well, maybe it's the accountant in me, but I don't want to go into debt. And CUNY colleges are normally very affordable. But the Zicklin School of Business, even though it's a CUNY college, it's well recognized and well noted. And I figured if I wanted a job at any of the big accounting firms, that would be the best place to go, especially that Baruch has an outstanding career program out there, and they're very great with most of my professors were coming from most of the big firms out there. My federal tax professor was a LLM at Ernst & Young, while my corporate tax professor worked at Deloitte & Touche. So I had it's like the opportunities at that particular college was outstanding, and so I know that I wanted to make the most of that.
Great question, thank you. Any other questions? Dave, yes?
I have a question for Nicholas. Nicholas seems to be a pro at applying strategy. What points do you have with writing your essay or going to an interview, speaking with them, to present yourself?
Okay, so the question is about pointers on the application process and interview process for graduate school as someone who's met some success.
Well, for the personal statement essay, I guess is what you're referring to. What I focused on, the fact that the vast majority of it was about what my research was on, my research experiences, and how what I knew about them, and how they helped shape me as an individual, and how they related to my overall goal in astronomy. Just for context, I like high energy astrophysics. And so a lot of my projects have dealt with high energy astrophysics, and I always related to the fact that I want to do this, and these are the steps that I took to get to this point. So you want to show a clear path in a clear direction. Even if it's not fully formed yet, even if you're kind of like, maybe I want to do this, but I'm kind of open to anything else, you still say, this is what I've wanted to do, and these are the steps that I've taken to get there, because it shows that you're serious. it also helps to have good recommenders. If someone can vouch for you, who's known in the field or who's known by the people at that school, that goes a long way.
Okay, we have time for one more short question. Is there one? Okay, going once, going twice. Let's thank our presenters. Very nice, and we look forward to putting you on the alumni lists for fundraising in the future. Okay, so we're going to have a student performance of a piece from Bonafide Women by Stephanie Ogaliza, Erlene Steffen, and accompaniment by Harvey Leona. And these are students who are working on some advanced work in the performing arts major. We're going to get a short performance from them, and then we're going to go to the poster session. But this is going to close out our lunch session. So let's welcome Stephanie Ogaliza, Erlene Steffen, and accompaniment by Harvey Leona.
After careful consideration and many sleepless nights, here's what I've decided. There is no such thing as a grown-up. We move on. We move out. We move away from our families and form our own. But all those basic fears, those basic insecurities, all those old wounds just grow up with us. And just when we feel that life and circumstance has forced us to once and for all finally grow up and become an adult, our mother says something like that. Or worse, she said something like that. We get bigger, we get taller, we get older. But without parents to defy, we break the rules we make for ourselves. Basically, we're all just a bunch of children running around the playground, desperately trying to fit in. We throw tantrums when we don't get our way. We confide in our best friends. We look for comfort where we can find it. And we hope. Against all logic, against all experience, like children, we never give up hope.
Who here has some money? Raise your hand if you have some money. Money? Who here likes the McDonald's? Burger King? I can give you the service. Oui. Bonjour. Je m'appelle Geneviève. Here I have $15. It's a small amount, but I did some services earlier. You see, I am a prostitute. And I look good. And I can give you better service than the Burger King and the McDonald's. Well, you sit here and you laugh at me and you do your face like this. I see you. I'm looking. Believe it or not, you know, this here is the Bible. I used to go to the church. Don't be offended. A long time ago, I went to the church and I wanted to get an anointing. I go to the church and I sit. Can I borrow this? I sit in the church every day in the front. I say, oh, Pastor, yes, thank you. Yes, Pastor, I listen to everything you say. Oh, yes, yes. And one day, I went to his office because he always said to make an appointment. If you have any extra, if you want to talk, or if you need any counseling, any guidance, I said, okay, Pastor, I make an appointment. I must make an appointment. And I went and I make an appointment, and my pastor anointed me. Anointed me. And I told my mother what he did to me. She said, Genevieve, you are lying, Genevieve. The pastor cannot do such a thing to you, Genevieve. send me out the door. She said, Genevieve, you are a disgusting girl. She thinks I'm lying. So, I go. I have nowhere to go, but I go to the street. Everybody doesn't choose a lifestyle that they end up. I did not choose to be a prostitute. There's only one man. After my mother kicked me out of the house, he's the only one that cared for me. He gave me clothes, he gave me the jewels, he gave me the shoes, he even paid for my hair. He did everything for me. My mother, she kicked me out. And so, I stayed with him. One day, after doing so much prostitution, these things, they didn't work too much, you know. So, I had a baby. And I didn't have a mother. I didn't have a family. I only had this man that was, you know, my pimp that was taking care of me. But I wanted something of my own. And I got pregnant, and I wanted to have the baby. And he said, if you have this baby, Genevieve, you cannot do prostitutes for me anymore, so you're not having this baby, Genevieve. And I had an abortion. Just sit and think about the small thing I just told you. Just a small story. Imagine what it's like to have a family and you express to them something that happened to you. And they don't believe what you have to say. And then they kick you out. And a stranger has to come and help you. And you feel that's the only person that you have that can help you survive for the rest of your life. And then you almost give birth to have your own. And they take that away from you. Just think about that. And now, prostitution, you can kiss my ass. But this time, it's for free. Au revoir.
I'm drinking wine and thinking bliss is on the other side of this. I just need a compass and a willing accomplice. All these doubts keep filling my head, cascading up and down again, up and down, around again, down and up and down again. Oh, I've had my chances and I've taken them all just to end up right back here on the floor. To end up right back here on the floor. Pennies in a well, $1 million in the fountain of a hotel. A fortune teller who says maybe you will go to hell. But I'm not scared at all. The cracks in your crystal, the cracks in your crystal ball. Sometimes you think everything is wrapped inside a diamond ring. Love just needs a witness and a little forgiveness and a halo of patience and a less sporadic pace. And I'm learning to be brave in my beautiful mistakes. Oh, I've held that fire and I've been burned, but I wouldn't trade the pain for what I learned. I wouldn't trade the pain for what I learned. Pennies in a well, $1 million and a fountain on a hotel. A fortune teller who says maybe you will go to hell. But I'm not scared at all. The cracks in your crystal, the cracks in your crystal ball. Irony, irony. It's hate and love, and what it does to me, and what it's done to me, and what it does. Pennies in a well, $1 million in a fountain of a hotel, a fortune teller and a black cat's cold stare. Walk on the ladders on my way to hell, I'll meet you there. But I'm not scared at all. The cracks in your crystal, the cracks in your crystal ball.
Sometimes I walk down the street, right? And people be looking at me like, why she look like that? People look at me like, oh, she got jeans on. Right now I got heels on, but most of the time I got my Tim boots on and my Yankee fitted, and they be like, oh, that chick is a lesbian. I was like, so just because I have jeans, Tims, and a fitted on and my hair up, I'm automatically a lesbian? So females can't walk around with jeans on, or boots on, or have their hair up on? No. I must be a masculine type of female, right? Wrong. I've been to the store with my homeboys, and that fool had the nerve to buy jeans tighter than these. Some of y'all might have them on. Some of y'all probably have on your wife's dress pants, but I'm not going to say anything. And I was just like, you know, that's not right, because we don't talk about you guys like that. So why y'all got to talk about us like that? I'm going to leave that alone right now. Y'all think about that. One day, I was in school, right? And I heard a group of, do you guys know what a cipher is? Yes? No? Anybody? A cipher is pretty much, oh, somebody knows, hi. A cipher is pretty much where a group of people get together and they usually like stand in a group and they usually like, they vibe and make music together. It's either rapping or singing or whatever kind of music you're into, that's what they do at a cipher. And I'm into hip hop music. So I'm over there, I heard the cipher, I'm like, I could rap too. And this boy was like, whoa, girls don't rap. Girls don't rap. Here we go again with the sexual discrimination. First I can't dress a certain way, can't put my hair a certain way. All of a sudden I can't do a certain type of music. I said, okay, so what is it that girls do? He said, well, y'all just put on the, you know, the really, really high heels. You get some really long hair, do some really nice makeup, and put on, you might get some fishnets if we approve. And make sure you have on a bra. Well, maybe not. So we're just pretty much props, right? That's what you're trying to say? I got you. So I was like, check it, right? Love, beauty, and sexual rapture. I'm your Aphrodite, want me now or after. I can hear with magic because I'm queen of the throne. I was teaching Isis, she was taking it home. Osiris took it down to the peoples at sea. Now every creature knows to be the Nubian queen, leader in the war, fighting battles for Kush. What makes you think your manliness can work with a push? Lay a kick or a slap, homie bust out the gap. I'm shooting you with Bible verses, pastor holding me back. You're supposed to be my face, baby, I'll be the pimple. You're supposed to be my areola, I'll be the nipple. Either way, you got my back, but you liking at that. I'm not just talking with support, I'm talking sexual act. You put yourself beneath the level. None no longer can go. Equivalent, I remember spitting in the man with the cloak. Yeah, Lukey chilling like a daisy with a smirk on his face. Yes, the dentist of the menace of the heavenly place. Ostracizing him to hell, had to give him his space. He was such a disgrace, he on the bottom of the waist, it's like he carried a tray. Imagine having what a period a year to date. I'm talking every single 365 a day. You wish your kidney stones was the only things in your way. Your style of switching better start. I'm even helping you pray, okay? I'm submissive when I'm needed, but I'm dominant too. How am I supposed to love if I can't get it from you? Love, beauty, and sexual rapture. I'm your Aphrodite. You want me now or after? Check that, homie. Peace.
Identity signifies complexity. Identity signifies reality. Identity signifies identity based on what? They say that you make the perfect lover. Look for no other. They say that you are kind and gregarious when all you are is conscious. They say that you must have skills because your record runs up high bills, boo. See, they say that you must never stop because now you on top. Who signifies identity when the bare of vanity is seriously in me? You know what they say about acting? They say it's sharing a bit of yourself that your friends and family don't normally get to see. But the problem with this, of course, is if you don't know yourself, then they don't believe you. They think you're lying. And I think that this has been my problem. I've been lying for so long, I don't even know the truth. I just throw myself into other characters, into other people. I like to say that I got lost in the shuffle, but then there's the kicker, isn't it? When I finally get on stage and I finally show me, all anyone sees is an actor.
Sometimes I shave my legs, and sometimes I don't. Sometimes I comb my hair, and sometimes I won't. Depending on how the wind blows, I might even paint my toes. It really just depends on whatever feels good in my soul.
I'm not your average girl from the video. And I ain't built like a supermodel, but I learned to love myself unconditionally because I am a queen. I'm not the average girl from your video. My worth is not determined by the price of my clothes. No matter what I'm wearing, I am always be in the Atrium.
When I look in the mirror, and the only one there is me, every freckle on my face is where it's supposed to be. And I know I created it and make no mistakes on me. My feet, my thighs, my lips, my eyes. I'm loving what I see.
I'm not the average girl from your video. My worth is not determined by the price of my clothes. No matter what I'm wearing, I will always be in the Atrium. No matter what you say, like a supermodel. 'Cause I ain't built like a supermodel. No matter what I'm wearing, I will always be in the Atrium.
Am I less of a lady if I don't wear pantyhose? My mama said a lady ain't what she wears, but what she knows. But I've drawn a conclusion. It's all an illusion. Confusion's the name of the game. A misconception, a vast deception. Something's gotta change. But don't be offended. This is all my opinion. Ain't nothing that I'm saying wrong. This is a true confession, a life-learned lesson I was sent here to share with y'all. So get in where you fit in. Go on and shine. Clear your minds. Now's the time. Put your salt on the shelf. Go on and lose yourself, because everything's going to be all right.
I'm not your average girl from the video, and I ain't built like a supermodel, but I learned to love myself unconditionally because I am a queen. I'm not your average girl from the video. My worth is not determined by the price of my clothes. No matter what I'm wearing, I will always be India or so keep your fancy drinks and your expensive minks. I don't need those to have a good time. Keep your expensive cars and your caviar. All I need is my guitar. And keep your pistols and your crystals. I really like a pretty piece of crystal. Keep your silicone, see I prefer my own. What God gave me is just fine.
I'm not the average girl from your video, and I ain't built like a supermodel, but I learned to love myself unconditionally because I am a queen. I'm not the average girl from your video. My worth is not determined by the price of my clothes. No matter what I'm wearing, I will always be India Ari. Sing. Hello everyone, good afternoon. That was just a piece of a show we have. We're going to have a bigger show with longer version of the monologues and the singing, and it's called Bonafide Women. This is what the flyer looks like. And the flyer will be around the school. It's free admission right here. Everyone's welcome to come. It's free to anyone. If you know anyone outside the school that doesn't go here, please bring them. And it's going to be May 12th, 2010 this year, from 6 p.m. to 8 p.m. on Wednesday. Wednesday, May 12th, from 6 p.m. to 8 p.m. And it'll be in the small theater. Remember, free admission. It's called Bonafide Women. And there's also another show, May 6th, we are the Drama Club, and there's a show before this show. May 6th, we're doing the complete works of William Shakespeare, and we have 3 of our males from the Drama Club that will be performing this, and that again is free of admission, and that will be Thursday, May 6th, from 12 to 2 in the small theater. You are all welcome. If you have outside guests that want to come, everyone's welcome. And this is what they're fighting. So thank you so much. You guys are a great audience.